Hello, everyone. I think we're going to get started. It's my pleasure to introduce Jeremy Nance today. He's one of our new colleagues at UW, and we were very lucky to recruit him from NYU a little over a year ago. He's a professor in the Department of Cell and Regenerative Biology, and he recently applied to become a mentor in our genetics program. And as I'm sure you'll see from his talk, I think he's a great fit. He has been working with the C.elegans genetic model system, taking advantage of a bunch of genetic and cell biological approaches to study cellular and molecular mechanisms underlying different aspects of embryo development, from polarization to gastrulation to different aspects of organogenesis. And since he started his lab about 20 years ago, Jeremy has successfully trained a very large number of graduate students and postdocs, and pretty much like all of them have done really well and have gone on to stay in science careers. A lot of them are faculty members. So I'm very excited to hear your talk, because you always give beautiful talks. Too fancy for me.
Okay, can everybody hear me? Good. Okay, so thank you for the opportunity. I'm excited to be here. This is my first full-length science talk since I've been here. I actually spoke in this room about 15 years ago, so I'm going to give you a little update on what's happened in the time being. And as Daniela said, the biggest change has been I am right across Henry Mall in Bach Labs. So several of you I've met and a lot of you I haven't. So if you want to talk about anything, you find some science overlap, please seek me out, send me a message, and I would love to talk to you. Okay, I have a slight change in the title. It's mostly what I wrote my abstract about. I was going to talk about two different projects, but I decided to make this all about one project and several different stories that kind of fit in that one area. Okay, so the big question my lab is interested in really is how during embryonic development organs first form. So lots of different cell types are in organs. They have to come together in meaningful ways. And perhaps the most dramatic example of this, and intimidating, if you ask me, is the human brain, where we have about 80 billion different neurons that have to come together in an organized way, make connections with each other for us to sit here and talk and listen to science. And so my approach has been, throughout my career, has been to take a reductionist approach to this problem and to try to boil down the developmental events that happen with individual cell interactions and how those might contribute to setting up an organ that's functional. And so one of the most important types of cell interactions that many of you are familiar with is the interaction between a stem cell and its niche. So the definition of a niche, and you'll see that this is pertinent for this talk today, is the environment around a stem cell, including cells and extracellular matrix, that instruct that cell to perform its developmental role. So that can mean regulating its quiescence, its division, its asymmetric division. And so today I'm going to be talking about a really simple stem cell niche interaction. It's 4 cells in the C.elegans primordial gonad. So the whole talk is going to be about these 4 cells and a support cast of cells, which I'll introduce in a minute, that play a really important role in their development as well. So the primordial gonad has 2 primordial germ cells. So these are the cells that are born in the embryo. And in the larval stages, when they begin to divide into germline stem cells, they make sperm or egg. And then there are 2 somatic gonad precursors, one for each of these primordial germ cells. And you'll see that they function as a niche and they give instructions to these cells in the embryo. Okay, so as I mentioned, the gonad in the embryo, once it forms, and I'll tell you how it forms in the next couple of slides, remains quiescent. So these cells never divide in normal embryo until the embryo hatches. And if there's food around, then these 4 cells begin to proliferate in tandem. And this is the adult worm, and this is the region of the worm that their progeny take up. So you can see they expand tremendously during larval stages, and they begin to differentiate into both sperm and oocytes. This is a hermaphroditic organism. So the same gonad makes both types of gametes. And so this begins, as I mentioned, only if there's food. And the primordial germ cells differentiate into what are called germline stem cells at this stage. So those are the proliferative germ cells that, that undergo eventually gametogenesis. Okay, so despite the simplicity of this organ, there's a lot of really, what I think is amazing cell biology that has to happen for it to come together. So the first thing that happens is the cells aren't born in the same part of the embryo. So the somatic gonad precursor cells here in yellow are born in the anterior, and they actually migrate to the germ cells. Those of you who know a little bit about germ cell biology, this is the opposite way that it happens in most organisms. So in flies and fish and mice and us, the germ cells actually migrate to where the somatic gonad is, and this is the opposite. I don't know why, but that's true. So then once the somatic gonad precursors reach the germ cells, they extend these membrane sheets that cover their surfaces, and I'll show you a picture of that, and they begin to wrap around them. And this is a behavior that's seen in the germline of most organisms and also in many different stem cell niche interactions, where a niche cell will make really intimate interactions with the stem cell. And there are lots of arguments about why that might be important. And I'll show you today some reasons that we've discovered. So then something really, really interesting happens, which will be the focus of most of the talk today. So now you're looking at the embryo, it's turned 90 degrees. So you're looking at one germ cell, even though it may look like two, the germ cell actually squeezes down in the middle and makes this large lobe-like structure, which pokes up into the third cell type I'll talk about, which is the endoderm. These are these blue cells, and the endoderm surrounds this lobe, and the somatic gonad precursor surrounds the cell body. And at the same time, a basement membrane first appears around this organ, which we'll talk a lot about. And finally, these lobes disappear and are digested by the endoderm, which is the last part of the talk that I'll talk about. So there's a lot happening in these four cells. A lot of interactions, both between the primordial germ cells and the somatic gonad cells, and also between the primordial germ cells and the endoderm. So this is what they really look like. So this is one of the PGCs. Its nucleus would be right here. These Mickey Mouse ears are the lobes sticking up into the adjacent endoderm cells. There are 20 of them here. And this is one of those somatic gonad precursor cells. You can actually see it wrapping here around the cell body. And in higher magnification here from an EM I took from John Sulston's lineage paper of the C.elegans embryo. This is that germ cell nucleus, the somatic cell, and these, the blue cells here are the endoderm wrapping around the lobes, which are right here. So there's all kinds of good stuff in the lobes, and we'll talk about one particular organelle and the consequence of this, the disappearance of the lobes on the germ cells. Okay, so in the first part of the talk, I'm going to talk about what the role is of this SGP wrapping around the PGC cell body. And so this was the work of a postdoc that recently left the lab, Dan McIntyre, who has his own lab now at the University of Virginia. And he was really interested in the question of why these SGPs wrap around PGCs, especially because this is a conserved behavior seen in lots of different germlines and stem cells, as I mentioned. So years before, we had actually imaged this process. And so here's a picture actually from the back end of the embryo showing the somatic gonad precursor cells just after they meet the germ cells beginning to wrap around their surfaces. And what Dan noticed is this happens right about the time that the basement membrane appears around the organ. And so he wondered what role the basement membrane might play. So here's a picture of that. So you're looking at the membranes of the SGPs. So the two primordial germ cells would be in these pockets here. And this is the gonadal basement membrane. It's like a little cocoon that surrounds this organ. Okay, so the first question, given the timing of its appearance, was whether the SGPs had a role in making this basement membrane. And then the second question Dan asked is, what is its function? Okay, so let me give you, I'm going to give you a little intro to some of the basement membrane proteins we'll be talking about today in the course of the talk. So there are receptors on the surface of the cell, such as integrins or dystroglycan, that bind directly to basement membrane proteins. And then the foundation of the basement membrane is really this trimeric protein called laminin. And laminin associates with the surface of the cell, and then other basement membrane proteins that bind to laminin can begin to organize. And those include things such as collagen, perlecan, and nitogen. So these proteins are found in all basement membranes, and then there are specialty proteins that are found in particular subsets of basement membranes, depending on the tissue or the developmental stage. Okay, so compared to vertebrates, C. elegans has a ridiculously simple basement membrane repertoire of genes, and in particular, laminin. So I mentioned this is a trimeric protein. There are alpha, beta, and gamma subunits, and they're unique beta and gamma subunits. So we can look at all basement membrane by labeling either one of these proteins, and we can take out basement membrane function by taking away either of these proteins. And then there are two different alpha proteins, epi1 and lam3. I'll talk about this one in particular. So you only have two different types of laminin in worms, which is far different from the complexity found in us, for example. Okay, so we're going to look at lam1 as a way to visualize the basement membrane as well as perturb it genetically. So this is a close-up view of one of the primordial germ cells. Its nucleus is here, and you can see laminin localizing around the gonad here. And also this is just between other organs. And what Dan did to address the role of the SGPs was take a laser and kill those somatic cells to keep them from migrating to the germ cells. And you can see specifically this gonadal basement membrane is gone, but the other basement membranes are fine. So the interpretation of this experiment was that the gonadal cells are required to template this basement membrane around the germ cells, and they lack the ability to do that themselves. Okay, so then taking away either the unique beta or gamma subunits, lam1 and lam2, what Dan saw was that compared to this nice array of cells in the control here, this is the nuclei, the PGCs are labeled in this case, and these are the two somatic gonad cells. All the cells are there, but they fall apart. So this wasn't a very surprising result because of what's known about basement membranes and other tissues. So the basement membrane is required for the integrity of these cells, but not for them to come together. And that was a really penetrant phenotype for either one of these different unique laminin subunits. Okay, so I mentioned there are two different alphas. It turns out that only one of those is found in the gonadal basement membrane, and that's epi1. And the combination of these two is found in many other basement membranes together. So we took a look at epi1, and it has the same phenotype as the other laminin subunits, but lam3, the one that's the alpha that's not in the gonadal basement membrane, has no phenotype. So this actually gave us a tool to more specifically disrupt the gonadal basement membrane as opposed to all basement membrane in the animal. And this becomes important because the embryos don't continue to develop if you take away all laminin, but if you take away epi1, they actually make it to larval stages. So that'll come up later. Okay, so what then? So the phenotype that we were seeing suggested that the SGPs may be holding on to the basement membrane in some way after they are required to nucleate it. So we started looking at receptors on the surface of the cells. And one I'll tell you about, dystroglycan, which binds directly to laminin, is found on the outside surface here of the somatic gonad cells. So this cyan color here is our SGPs. So you can see it surrounding it. And when we take away dystroglycan, you get exactly the same phenotype as you do with laminin. So they fall apart. Okay, the surprise came. These early results were not surprising. The real surprise came in these studies when Dan noticed a phenotype we had never seen before. And that is that we saw more than two primordial germ cells in the embryo. And that's these purple bars here. So in any of these mutants, there are extra primordial germ cells. But interestingly, in dystroglycan, which also disrupts the basement membrane, there aren't. So this tells us that laminin is playing some more specific role in regulating the number of primordial germ cells, and it's not just the integrity of the gonad that's important. And this resulted from a loss of quiescence. So the cells began to divide again. You can see that he captured this here in time-lapse video. In a normal embryo, they remain arrested in G2, so they're re-entering the cell cycle when you take away laminin in these guys. So that told us that the basement membrane was functioning as a brake to keep the cells from proliferating inappropriately in the embryo. So some beautiful work from Judith Kimball's lab here in the larval gonad showed that the Notch signaling pathway was what controlled the proliferation of germline stem cells in the larval gonad. And her lab identified direct targets of Notch in this capacity. And one of those genes is called Sigil-1. And so we took a reporter construct with the Sigil-1 promoter driving GFP to look at, to ask whether Notch signaling was inappropriately becoming activated in these primordial germ cells in the embryo. And that was the case. So if you take away the basal membrane here with either LAM1 or EPI1 RNAi, you can see this large increase in the signal of this reporter in the embryonic PGCs. So normally this signaling pathway is off, and when you take away the basal membrane, it turns on. And finally, we could use temperature-sensitive mutations in the Notch receptor called GLP1, and that nearly completely suppressed this proliferation. So just to put all this part of the story all together, what we thought at this point was happening is the basal membrane in the embryo would normally function as a brake to keep Notch signaling off in the primordial germ cells and keep those cells quiescent. And somehow, and we still don't understand how this works, the brake must be relieved when the embryo hatches into a larva and the germ cells need to start proliferating. Okay, so one conundrum of this so far is that the basal membrane doesn't contact the PGCs, but I just told you that Notch signaling went up in the PGCs. So we thought that maybe the basal membrane signal had to be relayed through the somatic gonad cells to the primordial germ cells. And to address this question, Dan used a laser again to kill the SGPs and take away the basal membrane with laminin, mutants, or RNAi. And he saw that this increase in the Notch signaling reporter largely was lost. So this suggests that the signal from the basal membrane is being relayed through somatic gonad cells to the primordial germ cells to keep them from dividing. Okay, so next we wanted to know what components of the basal membrane and also what receptors on the surface of the SGPs were responsible for this regulation. And so the first new group of proteins I'm going to tell you about are the integrins. And again, C.elegans has a really simple genetic repertoire of integrins. So this is a heterodimer that sits on the surface of the cell, directly binds to basal membrane components, including laminin and several others. And it has alpha and beta subunits, and there's a single beta subunit in worms called PAT3. So we can take away all integrin function with this mutation. And there are two different alphas. So you can only have two different integrin combinations in C.elegans. And if you look at the distribution of PAT3, it's a little dim on this screen, but it is, trust me, it is here on the outside of the somatic gonad cells, as well as many other cells in the embryo. And so Dan looked at PAT3 mutants, and they didn't have a proliferation phenotype. But then he thought, well, maybe they're functioning in downstream of laminin, and laminin is inhibiting integrin signaling, in which case they would function as suppressors of the loss of laminin. And so before I get into those experiments, he wanted to make this proliferation assay a little more robust. I told you that the EPI1 animals live longer into larval stages. And in fact, if you let them live several days, almost all the germ cells begin to proliferate in almost all embryos, and he would get up to 8 or 16 germ cells instead of the 3 or 4 that he was seeing earlier. And so what he did is take away laminin and then use, he took away laminin with this EPI1 RNAi, and then individually the beta subunit, one of the two alpha subunits, and saw that the PAT3 mutation almost completely suppressed this proliferation of germ cells, and so did this particular alpha mutation, but the other alpha mutation had no effect. So our interpretation of this is PAT2, PAT3 heterodimers of integrin were important for this laminin-mediated break on proliferation. So then to address whether these proteins were functioning in the somatic gonad cells, we needed to get rid of them. And this stage of C.elegans is a real headache for getting rid of proteins in a tissue-specific way. And several years before, we had developed a genetic method that relies on an endogenous worm protein degradation pathway called the ZIF1-ZF pathway. And so we took advantage of this to destroy integrin signaling, particularly in the SGPs. So just really briefly, the way that this works, in case anybody is thinking about using it, is there's a small zinc finger. It's only like 35 amino acids, and an adapter protein that recognizes that in a cell or in a protein, brings this protein to an E3 ubiquitin ligase complex and tags it for degradation. And it happens really quickly, like in 30 minutes or so. once you express the ZIF1 adapter. So you can express ZIF1 in particular cell types and really quickly degrade whatever protein you want. And so that's what so Dan did. He repeated these experiments using a genetically engineered PAT3 ZF1 allele. And he degraded it specifically in the SGPs. And you can see this is the same suppression of proliferation that he saw when he took away PAT3 everywhere. So our interpretation of this is that PAT2, PAT3 heterodimers of integrin in the somatic gonad cells are required for relaying this break from the basement membrane to keep the germ cells from exiting quiescence, which I just said. So there's that. So laminin in this, our genetics is telling us this is inhibiting some basement membrane signal, which is then functioning through integrin to keep the cells from proliferating. Okay, so then we wanted to know what this was. So what is laminin doing to, what is it inhibiting in the basement membrane that's signaling through integrin in this case? So we started to look at other basement membrane components. And I already mentioned these, but I'm going to focus on this very interesting protein called perlecan. I'll introduce it in a second. There's one worm gene, it's called UNC52, that encodes for perlecan. And it is localized, again, pretty dim on the screen, but to the surface of the gonad here, and it's also in many other basement membranes. So perlecan is a heparin sulfate proteoglycan that binds many growth factors and also can directly bind to integrin, which is why it caught our attention. And we had two different alleles that we looked at. One is probably a null allele, it's an early stop. And the second more interesting allele is one that very specifically deletes the RGD sequence in perlecan that binds directly to integrin. And either one of these alleles, when we used it in this proliferation assay, was able to suppress the loss of laminin-induced proliferation of the germ cells. So including the RGD deletion here. Okay, so wrapping up this part of the talk, laminin is doing some really important things to this organ. The first is more of a structural thing. We think through dystroglycan, which sits on the surface of the SGPs, it's holding the organ together after the SGPs nucleate the basement membrane around the PGCs. And through a mechanism we don't understand, it's inhibiting perlecan, and perlecan is either directly or indirectly binding to integrin and signaling through integrin rather to keep Notch off. And so when you get rid of laminin, this signaling pathway becomes active, and that turns on Notch in the PGCs, and they start to divide. Okay. So I'm going to switch gears at this point in my talk. So if there are any questions about this first basement membrane part now, feel free to ask, or you can save it to the end. Anything? Yes. It does. So it can bind integrin. It can bind integrin. Perlecan can bind integrin. Collagen can bind integrin. So
all these are complex.
They certainly could be. Yeah, we do not understand the biochemical mechanism of this. I mean, one idea is that the conformation of the proteins are still there when you get rid of laminin. They're just very disorganized. So we've looked at perlecan in a laminin depletion background. It's localized there, but it's patchy. Maybe it has access to integrin in a way that doesn't happen when laminin is there. We don't know yet. Yep. Any other questions before I move on? Okay, so now we're going to turn our attention to these cells that are not part of the gonad, but are really playing a quite interesting role in their biology, and those are the endodermal cells. So there are only 20 cells, and the primordial germ cells only contact 4 of them, always the same 4, right here. And as I mentioned, they wrap around these lobes, and eventually the lobes disappear, and they're taken up by the endodermal cells. So we wanted to know how that happened, why it was happening, and the first hurdle we faced in this project was that this is what the worm is doing when that's happening, and they actually have to move to continue to develop. You can do some pretty crude tricks like blowing nitrogen gas across the embryos, but who knows what you're doing to them to keep them from moving. And so if you look at markers of the endoderm and the germ cells and you try to do a Z-stack, this is a disaster. So this is what it looks like. You can see just not much. So to start this project, we collaborated with the Advanced Imaging Center at Genelia Farm, who had recently developed a lattice light sheet microscope which could take about over 300 sections through these embryos in about a second. So it's like a fast shutter speed on a camera. Those are the same markers on that scope. And you can see that it just freezes everything in place. And so for the first time, we were able to watch how these lobes were disappearing and what was happening to them. And this was all the work of a former PhD student, Yusuf Abdu. And this was published some time ago, so I'm just going to give you a really quick summary of it, and then we'll move on to some unpublished data that we're doing currently with this project. Okay, so Yusuf, and in his rotation project was to try to figure out how these lobes formed, because I thought it was an interesting problem, how a cell went from a sphere to a dumbbell shape. And he quickly came to my office and said, there's something interesting. I see these fragments of the cells inside the adjacent intestinal cells. And actually, he was a very careful guy. He had this red and cherry marker, and he said, well, let me repeat this with a different marker. So he picked a GFP marker, and he did not see this debris. And it turns out that's because it becomes acidified as soon as it's taken up by the endodermal cells, and it kills the GFP signal. So he looked at these two markers actually for a little while before he finally came to me and said, I think this is worth pursuing. And we figured out pretty quickly why his GFP marker didn't show that. But you can clearly see these are fragments of the germ cells inside the intestine in an L1 larva. So this is right after hatching. Okay, so then he asked, well, what happens to this debris if you don't have intestine? And there's a really great double mutant in C.elegans that very specifically lacks the endoderm cells. That's the End1, End3 double mutant. And you can see that the germ cells still make lobes, but they're just kind of piled up here between the cells. So it's not as if the endoderm is pinching these structures off of the PGCs to help make them, but the germ cells are making them autonomously. We know this now. You can put them in a dish and they make these lobes. And the endoderm is responsible for degrading them. Okay, so then at this point, we couldn't tell from the resolution of these pictures whether the germ cells in this genetic background missing endoderm were making lobes and releasing them and they just were sticking to the surface, or whether something more sinister was happening and the endodermal cells were actually biting them off the surface of the PGCs. So that would be phagocytosis or biting was his question. And so he devised a couple of experiments to address this. He took these endodermless animals. So here are the lobes that they make that aren't eaten. And then he photobleached them and saw that they very quickly recover. So that indicates they're connected to the cell body that he didn't photobleach. And then he did the complementary experiment where he loaded the embryo with a caged rhodamine dye and uncaged it in one of the PGCs, and just focus on these dots are just metabolism of the dye. But you'll see when I play this that the uncaged rhodamine begins to diffuse really quickly across all those lobes and into the other PGC. So this said that the PGCs make lobes, they remain connected to the cell body, and that the endodermal cells are actually biting them off of the PGCs, not just phagocytosing them. Okay, so I didn't even know what this word meant when we discovered this. I had never heard it. It's a word that you see a lot in immunological research, because as I'll point out in a second, there are several pathogens and also immune cells that interact with each other through the process of trogocytosis. So this means nibbling, literally. So one cell taking a bite of another cell, as opposed to phagocytosis, which would be a cell swallowing something whole, like a cell corpse or another organism. So it involves a biting step that the phagocytosis does not. And it's being seen in a wider variety of different types of cell interactions when people are looking more carefully, several host pathogen interactions, but also trading of antigens between immune cells. It's thought to be involved in although this is a little controversial, in the clearance of synapses by microglia to prune the nervous system. And here's our example here in the worm. This is the developmental example in this here that we're talking about. Okay, so we recognized at this point that there weren't really good model systems for studying this problem, and we had a great system because it was genetically programmed. It was happening at exactly the same time, as opposed to many of these host pathogen interactions or immune cell interactions. and it was in vivo. And so we used genetics available in C.elegans to really ask two questions. And I'll talk about this in the second part of my talk here. I'll talk about these. The first is, why would the germ cells do this? Why do they make lobes and only to lose them to the endodermal cells? And we have a few answers, but we're nowhere near completely answering that question. And then secondly, I think more relevant to trogocytic events and other examples, what are the cell biological mechanisms of this nibbling? Like, how does a cell actually bite off a piece of another cell? You can think about a lot of things that would have to happen for that to occur. Okay, so let's start with the purpose of trogocytosis. So we started looking around to see what was in those lobes. I showed you some EMs, and one of the things in those EMs were mitochondria. So we labeled mitochondria specifically in the germ cells. And in this movie, this cell is one of the germ cells. This is the lobe, and you can see all of these germline mitochondria moving up into the lobe here as it forms. So new student in the lab, Aaron Schwartz, began to look at this. I put these slides are out of order. There's Aaron. And so he wanted to know if somehow PGC lobe trogocytosis was regulating mitochondrial inheritance in the germline. And so he started by taking a closer look at what was happening with mitochondria at higher resolution. And so this is right when the lobes are beginning to form. Here is one just starting to form. The nucleus is down here. You can see them begin to move up into the lobe. And then this is right when the lobes start to be trogocytosed. You can see that there are some mitochondria in the cell body. Many of them are in these lobes. And we thought that some of them were being degraded and some retained. So this population of mitochondria, let's just to think about it, these are the primordial germ cells. They're going to make all of this population as the founding population for all of the gametes that are made in the next generation. So it's a really special group of organelles, and they have their own DNA. So we thought that maybe they would be regulated in some way through this process. Okay, I already showed you Aaron. So his question again was whether this was trogocytosis was regulating mitochondrial inheritance. And so the first thing that he did was just take a look, and he decided he wanted to be more quantitative than just mitochondria, which is a rather vague term since they're fusing and coming together, and it's hard to count them. So he wanted to actually look at mitochondrial genomes as a proxy for a number of mitochondria. And he did that two ways. And this is the first way that he did it. He tagged a protein called TFAM, which you can think of as the histone of the mitochondrial genome. So each mitochondrion has several genomes. They exist in the matrix, and they are packaged up into these little puncta called nucleoids with the protein TFAM. So you can see where mitochondrial DNA is. And so this is the strain he made with CRISPR, knocking GFP into the TFAM locus. You get these little puncta, and based on our results that I'll show you, and also studies in the literature, each of these puncta probably has one or a few mtDNAs in it. Okay, and first thing he did was ask, are there mitochondrial genomes in these lobes that are being eaten? And sure enough, you can see here's two lobes that are just cut off. This one is probably the GFP is quenched already, but this one has probably recently been cut off, and you can see their genomes here. So trogocytosis is responsible for eliminating some, at least, mitochondrial genomes. And then he looked at the number of these puncta that he could resolve in the embryo and in L1s. And you can just see from this picture, there's a pretty serious reduction in the number of puncta. And in quantification, he sees more than a twofold difference between this would be right before the lobes are made, and this would be after they're normally gone because of trogocytosis. So Aaron was a very quantitative person, and this was not satisfying enough to him, and he wanted to know exactly how many were being lost. And so he developed this painstaking cell sorting protocol with the help of Malik Patel's lab at Vanderbilt. And he found that he actually had to have two markers in the strain, one green marker for germ cells to sort for. And remember, I told you the somatic gonad cells are wrapped around those cells. So he couldn't, there was a very small fraction that he could separate. So he had to sort against somatic gonad cells. And when he did the combination of those two things, he was able to get populations of pre-lobe formation PGCs and post-trogocytosis PGCs. And just to convince you that this is what he had, and then he used droplet digital PCR on these populations of cells to assay the number of mitochondrial genomes. So just to convince you that he had pretty pure populations at these two stages, this was the purity that he saw based on the GFP signal. And you can see that the L1 PGCs are much smaller because they've already been nibbled. And that's volume quantification is here. So these are almost completely pure pre-turgorocytosis and post-turgorocytosis PGCs. And he saw something very similar to what he saw with the TFAM, but now he could slap a number on it and say that you were going from 400 mtDNAs into PGCs to 200 because over this period. So we thought this must be due to turgorocytosis, but we needed to show that. And so I have to introduce one more thing to you here, one more tool, and that is a mutation that keeps the lobes from forming in the first place. So we discovered several years prior that the way that the cell forms this dumbbell shape to make a lobe is it actually assembles they're arrested in G2, but they assemble a contractile ring in the middle of the cell containing myosin. So here's the ring rotated around. That would be right here. And you're looking at it in one plane, so it appears as two dots right here. And that squeezes down and makes the lobe. Doesn't squeeze all the way like a dividing cell, but it squeezes most of the way, like a corset. And so we knew from work from other labs, there was an activator of myosin called NOP1 that didn't have a major role in cytokinesis, but it did in the germline. And so when we took that out, we showed in this previous study that we were able to block lobe formation in most embryos. So Aaron redid the sorting experiment in NOP1 embryos and saw that there wasn't a drop between these two stages that was significant. So our conclusion then was, not surprisingly, based on what I've shown you so far, is that this turgorocytosis is eliminating about half of the mitochondrial genomes from the germ cell and getting rid of them. Okay, so the last thing he did is he went on to ask, well, what happens after the primordial germ cells begin? So why would you want this twofold reduction? What happens when they begin to divide as germline stem cells in the larva? And he saw that they maintained this level of 200 throughout at least the second larval stage, when he couldn't do a sorting after this stage much later than this. And interestingly, in NOP1, he asked, well, if you have too many, what happens during these stages? And the remarkable thing is, even though you start with way more than you have here in wild type at L1, within one cell cycle, they reset to 200. So we don't know how this happens. We speculate it's probably a delay in the replication of those mitochondrial genomes, but clearly the germ cells are recognizing that they have too many, and they're adjusting their number really quickly within one cell cycle to this set point of 200 where they like to be. And interestingly, this is the same number of mtDNAs that have been found in fly PGCs, and depending on the study, also in mouse PGCs. It seems to be a number that they like to have based on the biology and maybe the size of the cells. Okay, so one function of turgorocytosis of the PGC lobes is to create this set point. So we think that the PGCs inherit too many mitochondria from the oocyte, and this event gets them to this level where they like to be when they're dividing as germline stem cells in the larval stages. Okay, so our hypothesis is then that these lobes are like trash cans. So they're discarding mitochondria. They're undoubtedly discarding other things. And we think that these, like the mitochondria, they may interfere with germline development. They want to get rid of them in order to be the best primordial germ cells and germline stem cells that they can be and get ready to populate the gonad. So some of the questions we're interested in now are the better mitochondria preferentially retained by the PGCs? And this is a question that graduate student Gregory Gunberg is addressing. And also, are other cellular components selectively discarded as well? So you might imagine that mRNAs could be eliminated in these structures that the primordial germ cells don't want, or maybe damaged organelles, for example. So is there more than mitochondria in the trash that's relevant? Okay, and lastly, I'm going to talk about, in the last few minutes, our efforts to understand how this enabling process works in the first place. And most of this data, I'm going to summarize a published study, but most of it's unpublished. And here's the published part. So in this study, when we first described the biting off of these lobes, graduate student Yusuf Abdu did a genetic screen to try to find regulators of this process. And he identified what we call the scission complex, and these are proteins that are found in they function in the endodermal cells, not the PGCs, to cut off the membrane. And so these should be recognizable to those of you who are familiar with vesicle endocytosis. So there is dynamin, which can cut membranes, a sorting nexin, and F-actin, which forms a ring right at the base, in the endoderm, at the base of the lobe neck. So this looks exactly like an endocytic vesicle being cut from the plasma membrane, so severing the membrane at this back end. And you can see here is endoderm expressed dynamin at a lobe neck right before it's cut off. And Yusuf used the lattice light sheet microscope to show that this complex associated with the lobe about 10 minutes before we could detect that it was being degraded. It was very transient. So we think they're coming in, cutting the membranes and leaving. But a big conundrum with this project telling us that we were missing something was that structural biologists will tell you that dynamin should not be able to wrap around 4 membrane layers. So this is not like vesicle endocytosis where you have 2 membrane bilayers, we have 4. So what are we missing in this case? So we turned to genetics and isolated a bunch of mutants that had a similar phenotype where we had persistent lobes in the L1 larvae when we shouldn't have. And so the overarching question was, could we identify genes that would explain this difference between turgocytosis and phagocytosis, things that had a specific defect in turgocytosis but weren't general regulators of phagocytosis? Okay, this is the project of Julie Manikas, a current graduate student in the lab. And here are some examples of the mutants that she's been working with. So this is what a normal L1 larva looks like. These are the 2 PGC cell bodies. This is the debris of the lobes up inside the endoderm. You can see that this is the endoderm marker in green here. Here's an example of a mutant. This is kind of dim, but here's a lobe right here. It's actually inside the endoderm. I can't tell from this picture whether this is connected or not. I think you'd need to have another assay to do that. And here's an example of a persistent lobe that's sitting up inside the endoderm as well. So she and Yusuf together isolated 9 different alleles in this mutant screen that had this phenotype. And I just wanted to put a shout out for C.elegans in this sense, in that it's so fast and easy to do whole genome sequencing and cheap that you don't even have to map mutants if you find multiple alleles in a screen. So you can just sequence everything and see if there's a gene that keeps coming up over and over in the results. And that's exactly what Julie found. She found that 7 of the 9 mutants were in one gene called RAB35, which I'll introduce in a second. We made the same premature stop independently in 2 different alleles, and the rest of the mutations are either missense in conserved residues or a splicing mutation. But just to confirm that we had the right thing, Julie used CRISPR to re-engineer one of these stop codons into wild type, and sure enough, she saw the same persistent lobe phenotype that she saw in her screen alleles. Okay, so RAB35 is important for some step in this process. And this is a really interesting protein. So RAB GTPases are small GTPases that regulate different steps of membrane trafficking. And this particular protein is thought to largely regulate recycling of proteins from recycling endosomes back to the surface of the cell. And it has roles in lots of different biological events, including phagocytosis here. And so it was actually identified in worms many years ago for its role in recycling the yolk receptor, which takes up yolk into the oocyte, which actually revealed a lot about how it was working. Okay, interestingly for us, it had been identified in 3 different studies for its role in phagocytosis. And so in these 2 studies, it was shown to take up cell corpses. And in this study, it was shown to take up a structure called an exifer, identified by Monica Driscoll's lab, that neurons release, and the neighboring skin cells take up these exifers and then presumably get rid of them. And in all of these cases, RAB35 was shown by these groups to function in the engulfing cell. So we had fully expected to see a phenotype in the endoderm, but first we wanted to know what step in this process RAB35 was regulating. We thought that maybe it would be involved in the degradation of the lobes once they were internalized. And so Julie used a FRAP assay to test this hypothesis. And It's very similar to the idea I already brought up, and that is if you take a persistent lobe and you photobleach it, if it's still connected, this is kind of dim, but you can see there's some recovery here. And if it's not connected, there's no recovery. The recovery is never full, but we see these very different curves where one flatlines right away and another one continues to fill with unfrapped protein from the cell body. And so this would be a scission phenotype because there's still a connection, and this would be a digestion phenotype because the lobe is detached from the cell body. And so this is the sorting Nexin mutant I already mentioned to you that is in this scission complex, and we already knew that it was required for scission, and she used that as a positive control. And you can see that in all cases, she got recovery of the photobleached lobes because they're connected to the cell body. But interestingly, in the RAB35 persistent lobe, she saw two different phenotypes. Some were recovered and some were not recovered. And this was a surprise to us, and it told us that it's performing dual roles in trogocytosis. It's involved in scission. It's also involved in digestion. So then Julie thought, well, maybe it's doing different things in different cell types. So we fully expected it to just be functioning in the endoderm, but she started to knock it out also in different cells in this group. So the first data I'll show you is from the endoderm, and we did this with the same protein degradation system that I've already introduced to you. If she created a ZF tagged allele of RAB35 and degraded it specifically in the endoderm, these green bars here are mutant phenotypes, persistent lobes. And you can see her engineered allele has a very weak phenotype, but then when she takes the protein out just in the endoderm, it gets much stronger. Not quite as strong as the null, but that could be for lots of different reasons. The surprise came when she started looking at the phenotype, and I'm just summarizing a bunch of her FRAP data here. But it was very different than the mutant. So she didn't see these cases where there was a scission defect. Everybody was cut, just wasn't being degraded, okay? So our interpretation of those results was that the protein was involved in degrading, but not cutting the PGC lobes in the endoderm. And consistent with that, she looked at the expression of RAB35 specifically in the soma, by tagging it, by using a split GFP approach. She tagged it with GFP11 and then expressed the complementing GFP 1 to 10 fragment in the soma. And these lobes right here have just been cut off, and you can see hopefully this enrichment of RAB35 in the soma. This is in the endoderm right around those structures. RAB35 is everywhere, but it's enriching around those lobes that have just been cut. Okay, then she got a surprise. She took RAB35 specifically out of the PGCs, and she saw a weaker phenotype, but it was very significant and it was different from her control situation. And what was really interesting is it differed dramatically from the endoderm phenotype. Now she was seeing the scission defect in about half of the lobes that she tested using the FRAP assay. So, and the localization of RAB35 in the PGCs, we looked at it the same way, specifically with PGC specific expression of GFP 1 to 10. It was just on endomembranes and weakly on the plasma membrane. So it really didn't tell us anything new about the protein. Okay, so putting all this together, I already showed you our previous work. We identified this scission complex of proteins that functions in the endoderm to cut the PGC lobes. And so now we can add RAB35 in the endoderm responsible for degrading those lobes after they've been cut. But then our surprise was that RAB35 has a role also in the PGCs. And in this case, at least part of its role is to regulate scission of the lobes. We don't yet know how that happens. And we also don't fully understand why you get a digestion phenotype as well when you take it out in the PGCs. So maybe it's interfering with the digestion process subsequently in the endoderm. So we're still working on teasing apart these ideas. So some of our questions are, I think this suggests that the PGCs may actively participate in their own cutting. One possibility is that RAB35 has been shown to associate with the escort complex, which can cut membranes in that direction. So that's a hypothesis we're pursuing. RAB35 could be required to target factors to the lobe membrane that are required for interaction with the endoderm. So we're testing that. Or it could be involved in polarizing the PGC lobes so that they always form in the direction of the endoderm, which is something that happens in wild-type embryos. Okay, so I hope I convinced you that this meager organ has a lot of biological secrets still to reveal. So the interactions between PGCs and its adjacent niche I showed you are involved in organ integrity. So that would be the wrapping of the SGPs and the assembly of the basement membrane. I showed you that basement membrane signaling through the SGPs actually was important for maintaining the quiescence of the PGCs. And then finally, that this trogocytic event from the endoderm was regulating PGC mitochondria and probably other things that we haven't yet identified. Okay, I thanked people as I went along, but here are the people who did most of the work. Julie, still in my lab. Dan, again, with his own lab now at the University of Virginia, and Aaron, who just recently graduated. And these are my current lab members. And I mentioned we collaborated with Malik Patel and Nikita Saiba, his student at Vanderbilt, for developing the cell sorting protocol. And thanks to these sources for funding. And I'd be glad to take any questions. Yes.
So that your mutant lab certified, the mitochondria retained inside of the lobes, and it's what's happening there?
Yeah, what we know is that, so probably we haven't looked at that directly. That's an interesting question. Oh, I'm supposed to repeat the question. Sorry. The question was, are mitochondria retained inside these lobes that aren't digested in the RAP35 mutants? I don't know the answer, but I'm willing to bet yes. What we see in a normal embryo is that a marker for PIP2 rapidly disappears from the endodermal membrane, and it doesn't do that. So it looks like those are arrested at really early stages of degradation. So it would really surprise me if the mitochondria weren't in there, but we haven't looked at that directly. So it's worth testing. Yes.
I was just wondering, is it possible that your phenotypes could all be explained if like the lobe goes into the endoderm and then you have this double lipid bilayer, but if the process had to happen in a certain order, that the RAD35 first needs to pinch off the inside, the PGC membrane, and then the dynactin actin ring has to pinch, and the digestion can only happen after it's completely finished.
Yeah, that's a great question. I will try to summarize it, and I think this is exactly what we're thinking. So the question was, is there a particular order of events that have to happen in order to get decision? Maybe the PGCs cut their own membrane first, and then Dynamin could actually seal the deal, because now you only have 2 membranes to resolve instead of 4, and then they get digested. Clearly, the digestion would be dependent on the scission, so that's been shown for other things. So that would make sense. But I think I'm thinking exactly what you are, that if the PGCs have an active role, maybe the membrane is getting closed down and narrowed, perhaps by actin, for example, and that's in that ring in the scission complex. And then maybe there is a communication between the cells. The PGCs cut their membrane, and then that triggers the endoderm to seal it. I think that's a really attractive hypothesis that we're looking at. Julie's directly testing that. Yeah.
So I have 2 questions. First, what is responsible for transporting the mitochondria from the PGCs to the lobe? And you did show that some mitochondria are still retained in the PGC. So what exactly is regulating that? And my second question is, what happens when you perturb chromocytosis? That is, the excessive number of mitochondria, like, are there any mechanisms that you think are responsible for bringing it back to the ideal number in the progeny?
Yeah, so great questions. I'll summarize the first one first. Let me tackle that. So how are the mitochondria getting into the lobe, and how are they coming back before a subset coming back? You can ask Gregory these questions in about a year or 2, because this is his thesis project. We don't know that the original movement of mitochondria into the lobe, that could totally be passive, because what I didn't tell you is that the PGC is actually polarized when they make lobes. The nucleus migrates to one side of the cell, and then the lobe always forms on the other side of the cell. There isn't much space around the nucleus, so that could just be a mass action redistribution of mitochondria. We don't know. But we do know that the fraction of mitochondria in the cell body increases twofold right before the lobes are cut. So that's very strong evidence that there's active movement of those mitochondria. And Gregory's actually taking movies of this, watching the mitochondria move back. They look like they're stretched out, like they might be migrating along microtubules or something. So that we don't know the answer, but I do think there's going to be some kind of active migration back, which obviously raises the question about how are the ones chosen, if they're chosen, they're going to come back in. So that's a hypothesis we're really interested in. And the second question was, what happens when if you don't have turgorocytosis and you have too many mitochondria? So those were the NOP1 experiments. So that when the primordial germ cells don't get rid of their mitochondria, they have too many, the germline stem cells have too many, and they reset within a cell cycle. We don't know how that happens, but a logical hypothesis is that this is right when they start replicating their mitochondrial genomes. We do know that. I bet that's delayed, and they wait until they get to the right number. So that would imply signaling between the mitochondrion and the nucleus to coordinate the stoichiometry of those two things.
One more question.
Okay. Yep.
So if you said that the mitochondria increases twofold before it gets exported by the lobe. So if you somehow decrease the twofold increase in the mitochondria number, do you think that would affect the lobe formation in any way?
I don't know. We'll have to wait and see if we can figure out how they're coming back and we can block the return of the mitochondria, then we could ask a question like that. So we don't know if there's any connection between the mitochondrial movement and the cutting of the lobe. You wouldn't want mitochondria in the lobe neck getting in the way, certainly, of the scission happening, but we don't know if there's a regulation there. So Jeff, yes.
I'm trying to think about what reducing the number of mitochondria does for a PGC. You know anything about, I don't know, like reactive oxygen species or other things like that?
We did look at that. So we just kind of superficially, we use mitosox to look at reactive oxygen, and they're blazing mitosox levels in the PGCs relative to every other cell type in the embryo, which is something I never understood. Like, it seems counterintuitive that you would want to have potential damage happening in your primordial germ cells. So there is that. We don't know the basis of that at all. And what else was I going to say about that? So we haven't looked in larvae to see if that changes when the germ cells start proliferating at all either. Did I answer your question? I'm missing something.
Yeah, can I just ask a follow-up?
Yeah.
So given what do you know about
Oh, yeah.
programmed cell death in the PGCs, and like if you reduce the mitochondrial titer, maybe cytochrome C levels will go down? I don't know. I'm just.
We haven't. Okay, the question was about if could there be a connection between mitochondria and programmed cell death, because many of the germ cells in the germ line die through programmed cell death. What we do know from Jim Priest's work is that when a cell is dying in the germ line, it actually extrudes its mitochondria into the common cytoplasmic core, and they travel towards the oocyte. So they're actually not being destroyed through the process of apoptosis, but we have not looked to see. Part of the problem is in the NOP1 mutants, they correct right away. So we don't really have a way of having too many mitochondria for more than a single cell cycle to ask that question. Do we have time for one more or no?
Last question.
Okay.
How about your indulgence? Is it alternate segregation?
So the question is, what is there ER in the lobes? We have looked at ER. We have not looked at Golgi. The ER is filling that lobe. We haven't done movies to see what would happen, but I would be very surprised based on the EMs and our observations if there wasn't some endoplasmic reticulum that was also discarded.